Good afternoon, everyone. I'm Dr. Swati Chitrangi. Thank you for joining today's session on ReadyCell barrier models, predicting human drug absorption in vitro. Today, I walk you through how advanced in vitro models are helping predict permeability, transport, and safety in drug discovery and development. I currently serve as Head of Product Management at Mitox at Christian Biosystems. Christian Biosystems is a global leader in ELISA manufacturing and a distributor for advanced in vitro models, antibody, antigens, proteins, etc. in India. I have been working in life sciences field for over 16 years, focusing on drug discovery, admin science, and product management. My core area of work is developing and bringing to market advanced in vitro technologies, particularly humanized models and regulatory compliant assay platforms that support pharmaceuticals, biotech, and academic research. By training, I hold a PhD in biological sciences along with executive business training from IIM Lucknow. Over the years, my work has spanned 3D cell culture systems, organoids, in vitro toxicology, and translational ADME platforms. I have had the opportunity to lead teams and projects at Advances Group, Yashraj Biotechnology, and IIT Bombay, and carry out internationally recognized research at AIST Japan. In addition, I regularly share insights at industry forums such as CPHI, AMAPAC, ISSCR, and AIDS Delhi. I am a strong advocate of non-animal, human-relevant testing models in line with global regulatory frameworks like the FDA Modernization Act 2.0. I have also been fortunate to publish my work in peer-reviewed journals, including Stem Cell Research, Journal of Tissue Engineering and Regenerative Medicine, BMC Cancer, and Toxicology in Vitro. Today, I'll walk you through how advanced in vitro models are helping predict permeability, transport, and safety in drug discovery and development. During the presentation, you can write your questions in the chat box. I'm excited to be here today to discuss how ready cell barrier models can support, predict drug absorption, transport, and safety in drug discovery. So this is the agenda for today's talk. Introduction and objectives, then what are the challenges we face in the drug discovery process, intestinal permeability and bioavailability of the drugs. Then we will discuss the regulatory guidelines, example biopharmaceutics classification systems, then in vitro models and reproducibility in drug discovery process, then how in vitro model can be extrapolated to in vivo studies. Then in detail, we will discuss about the ready cell barrier models, that is Caco-Ready, Caco-Goblet, Prediport, Preditech models. Then along with this, we will discuss the transport studies and applications. Then we will discuss the case studies, including GI toxicity, inflammation models. Then in short, we will see the ready cell product portfolio. And at last, I will address the question and answer sessions. So as we are aware, drug discovery and development process is a very cost and time incentive process, which is divided into three parts like preclinical studies, clinical studies, and approval phase, where the drug is get approved by FDA. So to bring one drug in the market is cost approximately $2.6 billion, and it takes time of approximately 10 to 12 years. So it's a very costly and time intensive process. And in the meanwhile, due to the failure of the preclinical study or clinical studies, many drugs got are treated because of the failure of the studies, or maybe the preclinical studies is not getting mimicked in the clinical phase. So because of these, drugs get failed during the process of the development. Drug discovery process is divided, as I discussed, that it's divided in three parts: preclinical, clinical, and regulatory approval. So 90% of drug candidates fail due to poor prediction of human pharmacokinetics and safety studies. So we need very reproducible preclinical models to address these problems. One of the first barrier in drug success is the absorption. So any drug which we take orally administered drug, hindered by harsh gastrointestinal environment and defensive intestinal barriers. So as you can see in the picture, there are two types of barriers. First is biochemical barrier and second is physical barriers. So biochemical barriers, like there are enzymes in saliva, stomach, intestine. Like in saliva, there are enzymes like amylase and lipase. Then in the stomach, it's pepsin, cathepsin. Then in intestine, trypsin, chymotrypsin. All these enzymes, they act as a barrier for drug absorption. And in the physical barriers, there are enterocytes, goblet cells, am cells, nape cells, mucosa, then submucosal layers. So all these play a role of barrier in drug absorption. So, intestinal mucus layer, which is rich in negatively charged mucins, the epithelial is a tight junction, and the metabolic enzymes are there to support these drug metabolism and permeability of drugs. Then there are multiple transporters available in the intestinal layer. So it's efflux transporters and uptake transporters are there to form the barriers for all oral drug delivery. Drug bioavailability. So, drug bioavailability depends on many factors like physicochemical, physiological, and dosage factor. And there are multiple, as I said, there are multiple transporters involved in this process, like transcellular carriers, passive transcellular uptake, efflux transporters, and many type of cells are also involved in this process. And drug is absorbed via passive diffusion, carrier mediated uptake, paracellular transport. So all these plays a critical role in drug absorption and bioavailability. So there are some factors which mainly influence the drug bioavailability. Like key challenges are and the facilitators. So and these are intestinal permeability, which can be of two types: passive diffusion, active transport, efflux transporters like ABC transporters, solute carrier family transporters. Then there are some physicochemical properties like solubility of the drugs, molecular size, lipophilicity, all these plays a critical role in drug bioavailability. So, biopharmaceutical classification system, which has been introduced in 1995 to predict the oral absorption and bioavailability, aiding the drug development and regulatory approval. So, these are of four classes. First is high solubility and high permeability. Second is low solubility and high permeability. And third is high solubility, low permeability. And fourth class is low solubility and low permeability. So BioWaiver is a regulatory approval process where clinical bioequivalence studies are waived if a drug is high solubility and high permeability. Under the BCS, that is Biopharmaceutical Classification System, framework, predicting good in vivo absorption. It saves a lot of drug development costs and time, this BioWaiver studies. So in vitro research reproducibility. So 50% of preclinical studies are irreproducible, costing the industry billions and eroding the trust in experimental data. So the most of the preclinical models are failed to reproduce the results. This may be due to the contamination, cell ID, or the characterization of the cells, then the coating material which we use to grow these cells, then media, plastic wares. If there is change in plastic wares, then it will cause the irreproducibility of the preclinical models. Then there are how the media have been changed, whether it's partial or full media change. All these factors add to the irreproducibility of the preclinical model. So ready cell are the plates which are ready to use plates. It offers a consistent result because of its stringent QC criteria. So first model that I will discuss is CACO ready plates. So what is CACO ready plate? So it's used for in vitro permeability testing for drug development. So CACO2 cells are monolayer cells which are human colon adenocarcinoma cells which differentiate in 21 days in the in vitro plates, and it mimics the intestinal epithelium. And CACO2 cells are widely used to predict drug absorption but are mainly reliable for passively transported drugs due to variable transporter expression. You can see that the CACO2 cells, which is yellow in color in the plate in the picture, so it is grown on the transwell inserts, and there are two chambers. One is apical and basolateral. So if we are going to test any drug for the permeability, so we will add the drug at the apical chamber on the top of the transfill, and after 24 hours the drug will get transfused to the basolateral chamber. So this is how we calculate the efflux ratio, the drug present in basolateral chamber divided by drug concentration present in apical chamber. So this is how we calculate the efflux ratio. And to check the CAPO2 monolayer integrity, there are two types of test. One is tear measurement, which is the electricity barrier of integrity of the barrier that is formed by the CAPO2 cells. And second is Lucifer yellow permeability assay. Lucifer yellow is a fluorescent dye which is added to the apical chamber of the CAPO2 cells, and when after 24 hours the lucifer yellow will permeabilize to the basolateral chamber. So by reading its absorbance by fluorometer we can quantify the lucifer yellow permeability assay. So there is a stringent criteria to for the acceptance of CACO ready. So tear value should be more than 500 ohms per centimeter square, and lucifer yellow percentage should be less than 0.7%. So if the your barrier capillary to barrier is compromised, so the lucifer yellow transfer will be high. So you can see in compromised barrier, lot of lucifer yellow is going towards basolateral chamber. Similarly, if the tear if it's compromised, so your tear value will be less. And the permeability assay is a time-taking experiment which takes 1 to 24 hours, and tear assay is a very quick assay which takes only 5 minutes. So compatibility, so these tests are compatible with all the drugs, and it's repetitively and reproducible assays. So how you will extrapolate the in vitro permeability is assay to in vivo studies. So for that, the PAP apparent permeability coefficient that you will get from the drug assay in vitro, you have to use this formula and transfer it and extrapolate it to in vivo studies. So like if the PAP value is less than 10 million centimeter per second, so the drug in vitro, it will be, the drug absorption will be low, 0 to 20%. If it's between 10 to the power minus 6 to 10 to the power minus 6, then the drug absorption in vivo will be medium, like 20 to 70%. And if the PAP value is more than 10 million, 10 into 10 power minus 6 centimeter per second, then the drug absorption is very high. That is, it will be more than 70 to 90%. So this is how we extrapolate our in vitro permeability assay report to in vivo study. So this is one case study where CACO2 cells and CACO-ready plates, that is ready to use plates from ReadyCell, have been compared. The data have been compared. So drug-induced gastrointestinal reactions are major cause for post-market withdrawal of drugs. Many times what happens, the drug comes in the market and because of gastrointestinal reactions, it came drawback from the market. So in the left side of the image where you can see CACO2 cells have been used, and in the right side of the image you can see CACO-ready plates have been used. So four drugs have been used for this assay. One is nadolol, which is a non-toxic drug, then vamopressor and quinidine, which is a moderate toxic drug, and gemfibrozil, which is a highly gastrotoxic drug. So here in right image, you can see vamopressor and quinidine is not showing any fluctuation, which is not giving the real image of the drug behavior. Only gemfibrozil, which is a highly toxic gastrointestinal drug, is only showing the toxicity. And here only cell viability assay have been used to check the viability of the cells. Whereas the right side of the image, where capillary plates have been used, you can see how verapamil and quinidine is showing toxicity at 100 micromolar of drug concentration. And two criteria have been used to check these cell viability. One is TR values and one is Lucifer yellow assay. So it gives the real picture of the drug toxicity, where varampressor and quinidine has shown mild toxicity, and gemfibrozil, which is showing the high toxicity, and nadolol is not showing any toxicity. Second model is Kappa goblet cells, which is used to study inflammation. So Kappa goblet cell is a better physiological model for human intestinal functionality than Kappa2 cells. Why? Because here there is a co-culture of Kappa2 cells and goblet cells. So goblet cells are mucus secreting cells. So it mimics the more better closely to the physiological model of human intestine. As you can see in the table, that the absorption and mucus secretion is same for human intestine and goblet, whereas capillary is only showing 100% absorption. If you will see the tier values, that is transepithelial electrical resistance, so the human intestine model is very close to capogoblet. So capogoblet is closely mimicking the human intestine model. So this is one case study where anti-diarrhea agent, that is gelatin tannate, have been used to study the protective efficacy of intestine. So in this study, Kappa goblet cells, which is a robust model for evaluating anti-diarrhea agents and barrier protective therapy, is used for this study. So this study was conducted for 1 to 24 hours, and the intestinal layer, that is Kappa goblet model, was damaged with E. coli. E. coli has damaged the integrity of the Kappa goblet barrier, and after treating these damaged barrier with gelatin tannate, it starts restoring the barrier within 1 hour. You can see in the image that how TR value and Lucifer is yellow, flux is showing the integrity of the barrier restored. So this is a model which can be used to study inflammation in vitro. So this is a good model to study barrier integrity and antidiarrheal agents testing and efficacy of the drugs, and safety of the drugs. So drug transporters and endogenous biomarkers. So these transporters play a critical role in drug absorption and permeabilization of the drugs. So in liver, there are multiple transporters like OATP B1, OATP 1B3, then OC1, and finally it got excreted through bile. In kidney, there are also multiple transporters like OC2, MET12, OATP1, OAT3, and it excreted through urine. So these are the transporters which plays a critical role in drug absorption. So these are the membrane transporters which plays a role in drug absorption, disposition, safety, and efficacy. These transporters are located in different organs like intestine, hepatocytes, kidney, blood-brain barrier. So these are the circled transporters which are the highly cited and studied transporters, like in intestine, BCRP, PJP. Then in blood-brain barrier, most studied transporters are PJP and BCRP. Then in hepatocytes, OATP, 1B3, MET1, these are the most studied transporters. Then in kidney, some transporters which is most studied is OCT2, MET1, PGP. So I will, in my coming slides, I will discuss about these transporters and how it plays the role in drug absorption. So efflux transporters, as I said, there are two types of transporters. One is efflux transporter and other is uptake transporters. So efflux transporters which reduce the intracellular drug concentration and may limit the absorption or increase excretion. So these transporters are like PGP, BCRP, MRP1, 2, 3, 4, 5, 6, MET1 and 2. And there are different substrates which plays a role for these transporters like digoxin, cyclosporin, then metformin, cimetidine. All these drugs are substrate for different transporters, and the key sites are like for different transporters, different key sites. Some are present in intestine, some are in kidney, some are in blood brain barrier. So different places these transporters reside. These are the two transporters which is being offered by ready cells. So wild type where there is no expression of any transporters, then prediport MDR1, prediport BCRP. So, and regulatory agencies like FDA and EMA from Europe, they have mandated the study of these transporters to assess the drug permeability, diffusion, efflux kinetics, drug-drug interaction, and other transporter-based clinical strategies. So these two transporter, MDR1, which is also called as PGP, and BCRP, I will discuss in detail in my later slides. So this is a case study which has been carried out in Europe in Bangalore. So what they have done, they have used four drugs: quinidine, prozosine, and dantrolene, rhodamin 123, estrone 3 sulfate, which is a BCRP substrate. So in quinidine, which is a substrate for MDR1, the efflux ratio of 383, 838, sorry, 838, which is which was in MDCK cells expressing MDR1 cells, dropped to 14 because of the vaромpracil interaction. Vaромpracil is a inhibitor of MDR1 substrate. Whereas in MDCK2 wild type cells, you will see that there is no reduction because this MDR1 substrate is not there. Similarly, in CACO2 cells, there is no decrease because CACO2 red cells are not expressing any MDR1 transporters. So there is no change in the efflux ratio. Similarly, prazosin and dantrolene, which is a substrate for BCRP, showed 80-fold higher efflux ratio in MDCK2 BCRP versus wild type. And in CACO2 cells, where there is no expression of BCRP, there is no change. Efflux ratio was 4 and 2 respectively. Rhodamin 123, which is a MDR1 probe, efflux ratio of 4 in CACO2 cells were observed. And astrotone 3 sulfate, which is a substrate for BCRP, efflux ratio is 7 and CACO2 cells, and it reduced to 1 when the BCRP was treated with interacted with inhibitor, that is fumitremorgan C. So this is how prediport plates, wild type, MDR1 and BCRP having used to study the interaction of drugs. Next is optic transporters. So optic transporters facilitate drug entry into astrocytes, hepatocytes, kidney and brain cells. So most studied transporters are OATP1B1, which is expressed in liver, then OAT13, which is expressed in kidney, OAT12 is expressed in kidney and liver. Then there are some other transporters like ASBT, CNTs, and there are different substrate like for OCT1 and 2 there is metformin. Then for OAT1 there is penicillin, antiviral drugs. So different substrates are available for these transporters. So I will discuss about three most cited and studied optic transporters, which is available in ReadyCell. So it's a Preditech Met1, which is expressed in enterocytes, then OCT2, which is expressed in proximal tubular cells, and in hepatocytes, the expression of OATP1B3, which is Preditech OATP1B3 plates. So this is one case study which is used to study drug transport by OCT1 Met1 and OCT2 Met1. So uptake of MMP plus at two concentrations 10 micromolar and 50 micromolar was studied, and transcellular transport of these drugs were studied at different concentrations 10 micromolar and 50 micromolar. Then the metformin, which is a commonly used diabetes drug, it's used to study OCT1 and MET1 interaction. So, and transcellular transport of metformin, which is a diabetes drug, is used to study 10 micromolar and 50 micromolar. So this study has been conducted. Then they have studied a wide range of drug concentration, that is from 0 to 2500 micromolar for metformin and MMP plus also. And they have also studied the difference of different types of pH, like from 6 to 7.5 pH, having used to study the transcellular transport, how the pH is affecting the drug absorption of these transporters, MMP2 and metformin. As same MMP2 and metformin at different concentration and time at pH different pH variation have been studied. So gist of this study is OCT1 2 uptake and MET1 efflux transporter work together to drive the vectorial transport of organic cations such as metformin. Metformin also showed no saturation as we observed in our previous slide that 0 to 2500 concentration was studied, but no saturation was observed. This is because there is a high capacity of OCT1 and MET1 systems in liver and kidney. And then they have in this study they have also studied these transport efficiency is pH dependent. So at lower apical pH, it enhances the MET1 mediated efflux of organic cations. That is because in the intestine, the pH is low. So it correlates that in vivo study in vitro. And double transfected MDCK models are the valuable tools to study drug disposition, tissue concentration, and elimination mechanism of cationic drugs. So this is how these models have been used to study the drug interaction, and the in vitro permeability and drug interaction studies is mimicked to the in vivo studies. So this is how ReadyCell ready to use plates can be used to study the in vivo in extrapolation of in vivo models. So this is the summary of the slides that CACO ready and CACO goblets, two models ready to use plates, can be used to study drug permeability. CACO goblet for studying inflammation of the drug if the drug is causing any inflammation. And there are some predicate plates like wild type, MDR1, BCRP, which can be used to study efflux and blood-brain barrier transporter studies. Then there are ReadyTech, which are HEC2 93 cells, which is used for uptake transporter screening, MET1, OCT2, or ATP1B3. So these ready-to-use plate offered by ReadyCell are very easy to use. You just, after receiving the plates, you have to liquefy the solid shipping media at 37 degrees centigrade. It is a patented technology in ReadyCell, which offered them to supply these plates globally also. So after liquidifying the media, you have to change the media. Just remove the media and add the standard Capo2 media or any standard media. And then you are ready to go and perform your assays. So if you need any details about these ready to use ready cell plates, you can contact me at this mentioned email ID. So now the platform is open for any questions. If anyone has questions, so we will check the chat box for this. Yeah, so in chat box there are some questions which will be asked by Neha.
Hello, I'm Neha Chakravarti. I'm the marketing manager here. So the first question is, in what cases can BCS based biowaivers save time and cost in development? So Dr. Swati, over to you.
Yeah, so it's a very great question, Neha, which we got from chat box. So these bioever study is a regulatory process where the four classes that I've discussed in my presentation, that BCS, Pharmaceutical Classification System, has classified the drugs in four types based on permeability and absorption of the drugs. So generally, the class one, which is the drug which is highly permeable and absorptive, is bio-waived. Means it is waived for the in vivo bioequivalence studies. So this bio-waiver mainly save the cost in drug discovery and development process by waiving the cost of in vivo a bioequivalence study.
Okay, thank you so
To
much.
unmute. What's the
Okay,
second question?
yeah, Dr. Swati. The second question is, which transporters, efflux and uptake, are considered most critical by regulatory agencies for ADME studies?
Yeah, thank you for the question. It's a very interesting question, actually. So, regulatory agencies like in US FDA, European EMA, and PMDA. So, these regulatory agencies have mandated the study of both efflux and uptake transporters because both plays a critical role in drug absorption. So, these transporters are like, as I discussed in my slide already, BCRP, MDR1, PGP, then OATP1B3, OCT1, OCT3, these all plays a critical role and it's mandated by the regulatory agencies to check these transporters.
Okay. Thank you, Dr. Swati. The third question is, how can transporter assays support drug-drug interaction risk assessment?
Yeah, so as in my slides, in my presentation, I have discussed how gastrointestinal toxicity have been studied using Capo2 plates and Capo cells have been used to study drug interaction. And there are some drugs like metformin, which is a commonly used diabetes drug. So and its inhibitor also like Vamapracil was an inhibitor for MMP drugs. So this is how these in vitro studies of drug interaction can be studied using ReadyCell plates, and which these data, the PAP, apparent permeability coefficient data that we get, we can extrapolate this in vivo studies. So this is how it's critical and important.
So, so thank you, Dr. Swati, for the explanation. So yeah, we are done with most of the questions.
Questions
If
and
anybody has some more, we can address that too.
can open the session, ask all to unmute. Yeah. Yeah, if someone has any question, they can ask online also. Dr. Odisha, do you have any question? Okay, thank you everyone for attending this session. Hope it was helpful to all and it will be helpful everyone to understand in vivo models and in vitro models, how in vitro models can be used to study in vivo models. So thank you for your time and attention. I hope this session was insightful for you all and this will give you an insights to improve prediction of absorption, transport and safety in drug discovery process. Okay, thank you everyone. Recording was